I realized a few years ago that basically what I've been doing in my career is trying to connect data up with theory. And that's not as simple as it sounds, because sometimes you have to torque the data, sometimes you have to torque the theory, make them talk to each other. And it turns out that all of my most cited papers are this exercise of connecting things up. That's what Landy Arnold 83 was like, and Arnold 83. Anyway, you look back at them. There's a PowerPoint presentation for a talk I gave at Oslo in 2008 on my website, and you can link to it from here. There's one of these slides on data theory connection has a link, and you can go there and see other examples of what I'm trying to do in this talk. This talk is also based on a paper that just came out in American Naturalist in June, so you can, I'm going to give you abbreviated versions of the data output and so forth, but if you want more detailed ones, you can get it there. All right, so going into the specifics of this talk, so what you'll see in the middle, the relevance for at this point in the course is that this is an example of using ALWI to try and illuminate a really complicated kind of morphological behavioral character complex. And what I see, and what some of you see when you look at that bird of paradise interaction, is that courtship isn't just like, doot, here's a display, the female looks at it, chooses to mate or not. There's a whole complex of things. I mean, there are multiple morphologies that are coming into play. There's very strict stereotypy in the way there's that static jumping around period, right? Static display, and then a period of close-up tactile interaction. So if you think about what's the female's receiving, complex visual, auditory, tactual stimulation. And in most cases, we just have no idea what's going on in the female's brain that's either transforming all of that into a decision to mate or not mate. But one of the most successful attempts to take a functional complex like that and try and look at its evolution was the so-called Lande-Fisher model. And so what we're going to look at is the general problem of trying to connect up this Lande-Fisher process, which is a model for the evolution of sexually selected traits and the preferences that drive them. And here's the basic argument we're going to look at, and that is that models of this Fisher-Lande process have been successful and certainly prolific. There have been 30 versions of this model just in the quantitative genetics literature. And it's sort of been, up to this point, more or less an exercise in model proliferation in the literature. And it's remarkable that how few examples of actual tests with data that have been conducted. And I think the problem is this connection, making the connection between data and theory. So what I'm going to show is what we call the phenotypic tango, which is a more general version of the model that incorporates two characters, or it could incorporate many, on both the female and the male side. And this, I think, helps us capture the functional complex aspect of the male display, and there must be a multivariate female analog of that. So the problem is that, the problem and not the problem is that we can evaluate the predictions of the model with simulations, even though we can't do them analytically right yet. And so the question then becomes, using those simulations, can we use this phenotypic tango model, this new multivariate model, not so different from the univariate one, can we use it to account for actual sexual radiations? And the iconic example of sexual radiation are the birds of paradise and their displays. This is a radiation in which many characters in birds of paradise are more extreme, show a greater range than the entire remaining radiation of birds. So it's really quite incredible. So if we can account for that, I'm thinking we can account for almost anything. Okay, so then we're also going to try looking at a subset of the radiation with the OU and Brownian motion models that you guys have been using, we talked about yesterday and today. And we're going to see if they can account for the BOP radiation, the bird of paradise radiation. And in particular, what we're going to be looking for is whether we can take the parameters of output in ALWI and use them to estimate the parameters that are figuring in the original theoretical formulation of the Lande-Fisher process. We're trying to one of the things we're trying to do is connect OWI, BM models in general up with the sorts of problems, the sorts of phenomenology we're trying to explain with birds of paradise. And then we'll reach some conclusions. We'll talk about the future. All right, so sometimes in the course we've got a lot of students that are interested in sexual selection, but I think a few of you are interested in this. So let me give you a little bit of background. There's a classic paper by Fisher, classic treatment of this problem in his 1930 book, and there's this so Fisher basically took the idea of sexual selection. You've got characters that are affecting the mating success, and sexual competition of males, usually, and elaborated a model. Like many of Fisher's models in the book, it's a verbal rendition, but none of us doubt that he probably could have spun out an actual specification of it. But at any rate, right in the core of his treatment in his 1930 book is this statement. I'll use my Ronald Fisher British accent, which is pathetic, but still it gives you a slight flavor. The two characteristics affected by such a process, namely plumage development in the male and sexual preference for such development in the female, must thus advance together with ever-increasing speed. This is Fisher's statement of runaway sexual selection in these two features, the male and female features. And this excited me as a graduate student, excited a lot of people, but we didn't get a lot of insight into the specifics of what Fisher was thinking about until Lande made an explicit model of the process in 1981. And so, boop, we're in Lande land. So what Russ tried to do was think of a scale in which we're looking at some male attribute. Here it is, just these thin tails, feathers coming off. This is the red bird of paradise. And so we have in blue, what kind of a distribution? Normally distributed distribution of this male feature. And then in red, we're showing the distribution of sexual preference for this feature. And so what we're imagining is each female in the population has a preference curve with a most preferred male. Let's say it's the link. Here's a female that most prefers this kind of guy. And we take those most preferred males and imagine that they're normally distributed. And here's that distribution of preferences shown in red. So then what Lande did is model the coevolution of these distributions, which is assuming a lot of constancy in these variance terms, comes down to looking at the evolution of the means of these distributions. So you've got a problem in bivariate. Is this my water, I hope? That's good. Question, Ken?
Were these preferences measured before Lande made the model, or were they just inferred?
Never measured. Almost never measured in this way to give you a continuous function. Still a problem today. All right. So if you look at the evolution of those two means, the mean of the male trait, the mean of the female trait, let me tell you a little bit more about the model first. So here are the characteristics of the model, the principal features. You've got a male ornament, like that little tail thing, which affects a male survival and is also via female preference contributing or subtracting from male mating success. So there are two components of fitness that are being affected here. And then you've got a female mating preference that is based on the ornament in question, but doesn't affect the survival or fecundity of the female. We're going to relax those assumptions in a minute. Now, this is really a general treatment because the ornament needn't be preference. It could be a feature of genitalia, it could be a pheromone, it could be lots of things. And then for preference, you're thinking of some female attribute often in her brain, but sometimes in her morphology, that's affecting mating success of that feature in the female population. calling that preference. There are various ways to model preference. I just showed you one quickly. We're imagining that both traits are heritable, and because of assortative mating and sexual selection on the males, they turn out to be genetically correlated. And we're not going to model that process. We're just going to assume that this genetic covariance is created by the process and maintained in some kind of selection drift equilibrium, selection drift mutation equilibrium. Okay. Now, in the original Landry version then, in the 1981 paper, for example, Lande retrieved Fisher's runaway, first person to do that. But he also showed that there was another equilibrium situation in which the traits would walk towards a stable line of equilibrium rather than run away with ever increasing speed from an unstable line of equilibrium. People forget that Lande found this walk towards equilibrium, and it's the one we're going to use in our modeling because it's the one that you're most likely get in parameter space. This one requires rather extreme parameter values. This one doesn't. This duality of equilibria has been found in each of the 30 derivatives of Lande's model. So it's always there, and it's always the case that the walk towards rather than the runaway is more plausible on logical grounds, just on terms of parameter values. Okay. Let's look at some of these elements in a little closer detail. And this comes back to features from our early part of the course. If we look at the ornament, remember it's the little tail feathers here, males in blue. We imagine that it's normally distributed. We imagine that it's under a natural selection stabilizing fitness function, right? And one of the width, the omega width of that function is going to be one of our parameters in the model. And we also imagine, particularly in the simulations we're going to run, that the population is finite. So we've got selection driving the population mean towards the equilibrium, and we've got sexual selection causing the males to balancing or counteracting this selection so that the male population can equilibrate at some distance from the optimum under natural selection. Okay? Now if we on top of that make the population finite, the population will not just be a steady line, it'll bounce around. And the smaller the population, the more it'll bounce around this equilibrium, which is a balance between sexual and natural selection. Okay. Are you calm? Are you calm? Okay, let's flip to we have to flip back and forth. I've been doing these models for a lot of time in our laboratory, and I find that whatever your gender, you've got to sort of take a deep breath and bounce back and forth. It's a little bit of a mental workout. We're going to flip over to the female. I'm not going to put a slide in. Now imagine the icons are red. We're talking about females, and we've got a red curve here for the preferences. What we're going to do is relax Lande's assumptions a little bit and imagine that there's a natural selection function on preferences, but no sexual selection. So in the female case, the distribution of preferences should equilibrate right under the peak, but the mean should bounce around by drift, right? So it's relatively simple to characterize the evolution of the female distribution. And that's our model. But if you're not as good in stochastic processes as Joe, this is a pretty hard stochastic process to solve. I can't solve for its, I can't even solve for its equilibrium, and even more difficult to solve for its dynamics. And so that's why we're going to simulate the model, particularly when we take it into multiple dimensions. It's probably solvable, but I just haven't cracked the nut or found a collaborator. Okay, so now we turn to the issue of of why haven't any of these quantitative genetic models, there are elaborations that have sexual conflict. They've got all sorts of features that have been added in, virtually never connected to data. And so the problem is, here you've got birds of paradise and a lot of other things out here. You've got these models for the evolution of one or more traits in two sexes. Why don't they ever get tested? And there's a simple answer to that, and that is nobody gets data from a whole radiation. The theory's making statements about how an entire radiation plays out on a phylogeny, basically, right? Or in a set of replicate population. So that's what you've got to do. You've got to get that kind of data and test it with a theory somehow. So that's what we're going to do. And this is why we have the models disconnected, because the people elaborating the models haven't been interested in finding or even thinking about the kind of data they need. And the format, right? How do you get this to talk to that? You've got to get this theory to play out its predictions on a tree, because that's where most of our data are. Sort of obvious thing when you say it. All right, so here are birds of paradise. Let's look at their biology. We looked at their displays for a minute. Now let's look at the features they bring to this exercise. First of all, you never get the phylogeny, right? Phylogenies evolve as you get more characters and more taxa and new methodologies. But recently, a time-calibrated phylogeny was published for birds of paradise. That's good. We know from the kinds of observations you just saw that there's male display of features like plumage and song and all sorts of things that are the basis of female choice. I mean, it's easy to imagine. We don't have data that female choice from that video is happening, but it's plausible that it does. There are morphological components of the male display, and there's data on the male morphology. Without even going out to New Guinea, we can find data in the literature. And what I was able to find in particular is data on wing length and tail length. So I'm just going to say, and remember when I said we looked at that static display and the males go, woot? You can imagine that there's, for everything going on, the length of the wings and the length of the tail are a component of that static display. So we're going to say, give that as our justification for just treating this as a simple problem in two morphological dimensions. We have data on tail length and we have data on wing length. There are all sorts of other things in play. The lovely plumes and so forth that you saw stick up. No, not incredibly, never measured by anybody, right? Songs recorded across the radiation, no. So I'm talking to Edwin Scholes and Tim Lamon, who are at the Ornithological Lab and have a big project on birds of paradise. They're psyched and I'm psyched to try and go beyond these two features. But for now, we're just going to illustrate the approach with two characters out of the many, many dozens as you could probably look at. Okay, so here's the recent phylogeny for birds of paradise. These are all outgroups. Turns out birds of paradise are just crows. They're fancy crows. They have a common ancestor about 23, 24 million years ago. And so we have a pretty well-resolved phylogeny. And as you'll see, when we get data, we've only got it for this one genus, Paradisia, up here, right? It has a root about 9 million years ago. So that's going to be our test case. And here are all the fabulous things they do. If you aren't a fan of birds of paradise, you can go to the Ornithological Lab's website and you can see displays by all these incredible displays. And all of, like here's the Saxony bird of paradise with these incredible plumes off its head. I thought I showed you. They don't even have Wilson's bird of paradise, the one we looked at earlier. At any rate, you could spend a lifetime modeling this genus, of course. And here's the justification for using the particular features we're using. wing length and tail length. Here's the length of the tail. And here you can see this female. I like the static display. You know, we have no idea what's going on in the brain, but it's plausible that the wing and tail are part of her mental evaluation. Okay, now let's look at the phenotypic tango. The particular version of the FLP that we're going to use in our simulations to evaluate its ability to account for the tango. 2 male traits, ornaments, 2 female preferences, selection on the female preference, finite population size. We're going to plot the current mean of the ornament distribution in a single population and the corresponding mean, bivariate mean of the female distribution as just 2 dots like this. But you can imagine a circle, a bivariate circular distribution, or it might be elliptical, right? This is the P distribution. I just did it in dotted lines. See. And we're also going to superimpose in this space multiple evolving populations at once. We can do this very easily in R. And I'm just going to show the bivariate ornament mean, the bivariate preference mean. I'm going to connect them with a solid thin line like that. The preference traits are preference. So, let me back up. That's a good question, Moniki. We have wing length when we do on this axis, right? Here is preference for wing length by the females, right? We have male tail length, and here's preference for tail length. And so it's just like those curves I showed you at the start, right, where we had the preference was a single curve. Now it's a bivariate distribution, just like a surface, if you like, and matching, and we can plot them on the same axis, right, because the units are the same. I'll say it again. Yes, trait of the male. But in the female distribution, if you go up to each female and say, how do you feel about males? Female might answer the question by saying, I like a male with this body length and that tail length, right? You ask all of the females in the population what they prefer. Take the distribution of their most preferred mates, and this is the most preferred male in the female. You got it? Yeah. Remember, we're doing simulations. We have complete control. Yeah, we can make the parameters of, the distributions be anything what they want. But once we let them evolve, then that plays out according to rules that are, our modeling assumptions. Okay?
Okay, but so the ornamentation have been measured on actual specimens, so you have some sort of starting values for those?
We're getting there.
Okay.
I'm going to show you the model, and then we're going to come back to the data, and I'm going to plot the data in these dimensions, and then we're going to run the models and see if we can account for it. Okay? Good questions. The other thing is I'm going to standardize the phenotypic scale so that we're in units of phenotypic standard deviation, so that one on this scale and one on this scale are one phenotypic standard deviation. And we have within population data, as you'll see. All right. Okay, so with regard to this model of the FLP, there's good news, and that is that the model's more general than past versions. A lot of naturalists hated Lande's original assumption that there was no selection on preferences. So we've adapted and made it bivariate, the most common assumption that there's stabilizing selection. That the costs on preference, if you like, are normally distributed. I've already told you there's no analytical solution, but we can go quite far without it for the time being. And as I'm saying, we can determine the model's behavior before we even do the fits by just doing simulations. So we'll do some simulations just to get comfortable in our Zen space with how the model behaves, even if we don't understand its properties in a nice analytical setting. All right. So I'm going to go to some simulations now. I'm going to go to my website where I put these simulations up for you to look at, for readers of the article to look at too. I'm just going to show you a couple of these. Come on, Kirksher, where are you? There you are. Just pull this down a little bit. So if you go there, you can read the captions off here on the right. All right, let's just start with a simple situation. Whoop! Why aren't you going there with me? I have to get out of PowerPoint. Come on. Okay, here we are. All right, maybe I hit something. All right, so let's start with let's just go back to the ornament, right? So here's the ornament in 2 dimensions. And the current value for the bivariate mean is at that blue ball, right? And so what we're looking at is the simple evolution, bivariate evolution of that bivariate mean in really wing and tail space under drift selection balance, right? So it is pulled towards the center by stabilizing selection, and it drifts away from the optimum under drift. We have an equation that tells us how much variances we'll expect among many replicates of this process at any time t, and this blue line shows the 95%, they're normally distributed, those replicate means, and this shows the 95% confidence limits for the process once it reaches its equilibrium. Now if you watch the start, let me just take a minute because that line was showing you the approach to the equilibrium when you start replicate means at the center, right at the optimum, okay? Whoop, and then bam, you reach that stable expectation very fast. All right, now let's do it with replicate populations. So down here in this corner, the optimum for the ornament is here. Here's the 95% expected distribution, and I'm showing 100 populations evolving under drift selection balance. Okay. Up here, 100 populations, the same 100 as down here, but these are the preference means evolving just under natural selection. Okay, and you'll see those when it goes back to start. You'll see those the 95 confidence limits expand out quickly as a thin ellipse till it reaches and then here I'm also showing the distribution in the and you can see those distributions are very close to the expectation. Those distributions that an empirical 95% ellipse that we can estimate. Okay? No sexual selection. It's as if the female traits are out there, the male traits are out there, but there's no preference being exerted. Now we're going to connect them. Now we're going to say that the preferences actually affect one component of fitness in the male, mating success, which isn't in the simulations up to this point. And when we do that, we find that the ornament means are pulled over towards the preference means. And notice I've made the preference optimum some distance from the male optimum. They might coincide. I made them different so you can see this effect of the preferences pulling the means. And you can see the struggle, you know, the dynamic struggle. The preferences are being pulled towards their optimum. The ornaments are being pulled towards their, what is that called in the Rocky Horror Picture Show? Dynamic tension or something like that. Anyway, there it is. All right, so another thing to notice, and this is the main point of this slide of the couple, before when we just had natural selection, you can take the size of the radiation that we'd expect from replicate lineages, right? So under just natural selection, we have a very, very small expectation for the radiation, right? It's going to be inside this little ellipse. But now when we have sexual selection acting on the ornament, the radiation is multiples of what we expect under sexual selection, right? Right? And that's good because that might be getting us the kind of radiation and morphologies that we see in birds of paradise. It looks extreme, and this is saying, yeah, it's going to be a lot more extreme than just, it's going to counteract the effects of stabilizing natural selection. So, and to accomplish that, basically we have to make selection on the preferences weaker than on the ornament. If it's the same size, then we'll get a radiation that's just the same size but in a different position, right? So to get the radiation to work, we've got to relax selection on the preference. And the question is, how much are we going to have to relax selection to explain the radiation? That's going to be our issue. And you'll notice that here we're in units of phenotypic standard deviation. I can't remember my parameter values, but they're realistic. And you can see we're only out there about, the limits, 95 limits, are only plus or minus about a 10% of a phenotypic standard deviation. This is a very modest radiation. And even with preferences exaggerating the radiation, it's still very modest. You can see we're only out there, not even a whole phenotypic standard deviation, right?
Should we relate that to like functional constraints and flight or something?
What, the natural selection? Yes, that's what we're talking about. That's right. Too big a tail, you fall off when you land. Too little, you can't fly fast. Something like that. Now, natural selection on the preference is a little harder to talk about, and I think we should defer that to the discussion because it's a bit of a mind torque. Yeah.
How much of that, if any, that increase in variance you see is due to reduced NE as a function of preferences, strong preferences?
Effective population size affects the width, right? Right. But it is the preference, selection on the preference, that blows it beyond the limits that you'd expect under natural selection, right? Effective population size affects these equilibrium limits. We blast beyond those with preference. So whatever this is a function of additive genetic variance and effective population size, whatever they are. And you can go beyond those limits, whatever they are, with preference.
Steve?
Yeah, Joe?
I'm just lost. Which slide are you on? I cannot see the slide.
I'm not on the slides. I'm on a website. So, yeah, I think I'm about, if you look, there are four, I'm not going to go back there because I'll lose this. There's a quartet of figures, and this is one corner there in that quartet, this particular slide. It's B. There's A, B, C, and this is B. Slide B. Yeah. Okay, we're going to go on.
No, I have a question first.
Oh, you have a question here? We're not going to go on.
Well, so I'm curious about the ellipse for the females being actually bigger. Do you think that's due to reduced selection on the...
It's because there's relaxed natural... We assume relaxed natural selection on the preferences, and that produces... Well, you'll see later in other figures, you can get... We don't have an expression for how big the radiation, how big the ellipse is going to be for males under sexual and natural selection, but basically they end up being larger than the preference expectation. All right? Let me illustrate this fact that the necessity of making selection on preferences weaker than on ornaments to get an exaggerated radiation of morphology. Let's just flip things. Let's make selection on the preferences stronger than on the ornaments, right? If we do that, we get a radiation that's, can you see it, smaller than we'd expect under natural selection, right? We've really got to have weak selection on the preferences if we're going to explain anything, if we're going to get birds of paradise. Also, if we make, the selection on the ornaments, the selection surface elliptical, that means that there's correlational selection where we've got selection that promotes a correlation between wings and tail, if those are our characters. That affects the shape of the equilibrium distribution, as does the shape of the bivariate selection surface that's acting on female preferences. Point is, it doesn't have to be circular. We can make almost anything we want within the limits of Landyland, right? All right, now we're going to go back to, I'm going to try and go back to my PowerPoint, recent. Not you, this one. See, here's the link that would have gotten you there, Joe. Okay, now we're back in the PowerPoint. Okay. So we have a model that that seems maybe capable of accounting for the radiation, but now we need some data to really see if we can produce the pattern of the radiation, male traits being more exaggerated than female traits. Can we account for the actual extraordinary width of the bird of paradise? We're going to need data to do that. And like I said, without collecting data myself, I found some data in the literature. But let me show you the radiation at first, though. We're going to have to focus in on just the genus Paradisia, which has got, 7 species in it and multiple subspecies, all on the island of New Guinea. And that part of the radiation plays out over 9 million years, which if you give like 5 years, I can't remember what I used, but at any rate, that's about 188,000 generations, using sort of a standard estimate for bird of paradise generation time. Okay. So that's the radiation we're going to focus on, and it's because Mary LaCroix, working at the American Museum, collected data on just this genus, and she measured, among other things, tail length and body length. And here I'm showing you not preference, but female tail length and wing length, and here's the actual data on the 14 populations that she measured of male bird of paradise. These are the species means. No correction for phylogeny, just the tip covariance. And you can see that's the sort of elliptical distribution of tail length by wing length that we could produce with our model if we wanted to. No. Each dot is a species mean, okay? Okay. Blue, male ornament, as before. Wing length, tail length. This is not preference, which nobody ever measures on a radiation, almost never. It's the female tail length and wing length. And you can see that the radiation of wing length and tail length is much bigger in males. I mean, confirming the idea that it's been driven to an extreme because it's participating in this display. So the other thing we get out of this is if you look at the width of this radiation, just say, well, how many phenotypic standard deviations is the radiation? If we take the 95% confidence limits as a sort of a measure, sort of standard statistical measure for how much radiation we get, just using TIPS data, we're accounting for about plus or minus 12 phenotypic standard deviations on either side of the mean in each of the two characters, right? 12 that way, 12 this way. So I think that's what I did. Yeah, 12 from the mean. Okay? So that tells us how much radiation, how much character evolution we've got to produce with our model in 9 million years. All right? Touch of reality. So the first point I've already showed you is that we can produce the pattern. The big question is, can we produce the extent? So to look at the extent issue, I'm going to look at, let me just stop and tell you what I'm going to show you. We're just going to look at one feature. I think it's going to be tail length. But in any case, we're just going to look at tail length evolution, just one feature in the males. And well, let me show you the graph. Let me show you the next slide, and then I'll make the connection. All right, here's a simulation with our phenotypic tango model. And what I've done here actually is just superimpose the evolution of the ornament mean and the preference means in a single dimension. and I've done replicate populations of the model. So it turns out the model is an elaborate OU model. And we can see that because evolution is bounded. You see it going out here to limits and sort of not going beyond that. So let me show you a few features on this model. Here's ornament and preference lineage means in units of within population phenotypic variance. And so you can see here's plus and minus 10, here's plus or minus 12. So we're going out far enough in our simulations. In fact, we're going out far enough in about 20,000 generations. Let me show you a couple other things on the graph. This black band shows us our expected 99% confidence limits for the ornament if we just had natural selection, right? Here's the 95% confidence limits for the preference. So it's much weaker than the selection here. And here you're seeing the actual lineage means out there. They're popped way beyond the natural selection limits and even beyond the limits that we expect under preference, right? In fact, if we take twice the 99% confidence limits, that's this dotted line, that does a pretty good job of accounting for the lineage radiations in ornament. Okay, see it? The other interesting thing is if you look closely at these lines, you can see that on this scale, the preference means are right on top of the ornament means. See how tight the coevolution is? Very, very tight. And actually, that's pretty robust under choices of parameters. It's mainly affected by the tightness of preference for the male traits. But it's this mirroring of the preference radiation by the ornament holds under pretty broad value. So here's the point. According to this model, we have a blindingly important insight. And that is, we have an empirical problem measuring preferences in multiple populations in multiple dimensions. This model is telling us that the ornament means is such a close mirror of the preference evolution that we might as well be looking at the preferences. Yes. Huge empirical shortcut if we can confirm that. And notice that we've got a pretty reasonable effective population size, right? 1,000. Okay, so here's the troublesome thing. Here's the omega value for stabilizing selection on the ornaments, 9. That's a width that's about 9 times the width of our standardized trait. But look at how weak we have to make the preference function. 900, I mean, it's really broad. And if you think about that, how are we doing on time here? Pick up the pace here, I guess. If we pick up, oops, where are we here? I'll come back to that point. I guess my slide's up ahead. So we've got a problem. So this suggests that what we might do, if you don't, the alternative way of trying to model this is either weak preference or let's have the preference optimum move, right? And so here I'm having the preference optimum move at a modest rate, and you can see I can easily account for the radiation doing that. We're still Ornstein, we're still, well, now we're Brownian motion. So I've got Brownian motion of the preference. So it's just going to, the problem in long time is we're going to produce a radiation in which the tail links are from here to the sun, right? All right. But on the time scale we're talking about, this is an alternative explanation for the radiation, according to our simulations. All right, so here are our conclusions for this fitting the model. We can get evolving preferences control the evolution of preferences. We can account for the patterns of ornament evolution with the model. But to account for the extent, we've either got to have really weak stabilizing selection on preferences or we've got to have the optimum move. And it doesn't have to move at any phenomenal rate. I mean, a really, as you'll see later, a modest rate of preference movement, optimum movement, just a little jiggle each generation. Just very slow Brownian motion of the preference optimum would explain the radiation. Now that, well, okay. Okay, now we're going to turn to standard Ornstein-Uhlenbeck models. Now the problem is that there's no package out there that simulates the process we're looking at. That is to say, one trait under this complicated kind of selection exerted by another trait. No package has got the, this kind of bivariate evolution model. It's just way beyond. I mean, it's not impossible, but nobody's done that yet. But for these simpler models, which we might apply just to the preference or the trait, we've got nice analytical solutions. When we estimate the parameters of OU or BM, we get parameters that confound inheritance, selection, and population size. So they're a little hard to interpret. We'll show you how you can do that, though. But we can account for phylogeny, and we can use likelihood to compare alternative models. So let's go there. Let's fire up OWI. And taking this, sitting in here like a participant, I thought, I'll use OWI. I'll use OWI to use this. I went back and used Brian's tutorial to learn how to use OWI. Took a while for me, but I got there in about a week. All right. So the next part, we're going to review the contrast between BM and OU model expectations. We're going to test model predictions on the bird of paradise tree, and then we're going to. I didn't have this. The version of OWI doesn't have continuous motion of the optimum, but we're going to use we're going to paint selection regimes and have one selection regime in which we change the position. We're going to have two regimes, and we're going to see if we need to account for a shift in the optimum. That gives us a higher likelihood. That suggests that movement of the optimum might be the underlying process. You with me? We're just going to look at ornament evolution. That's all we have data on with OWI, looking at the BM and the OU fits. And here's a nice takeaway. We're going to show how you can use this framework to estimate FLP parameters, things like omega, N sub B, well, omega and what else? We'll have to look at, I can't remember what else we're estimating. All right, so here's our review. With BM, you get an ever-expanding evolution. That's what's going to happen when we let the optimum for the preference be under BM. Or you get bounded evolution if you hold the preference optimum constant. You're going to get those two patterns. Here's our tree. Here's Brian. We have so here are our 14 taxa. We're going to let this portion of the tree, which is a sort of more recent proliferation, have a different selection regime here shown in red, right? I didn't paint the edges or branches red, but that's what we're talking about. We're not going to say what the optimum is. We're just going to say let's imagine this is a separate regime, and then we'll do our runs and fit the models. We had Mary LaCroix had estimates of within population variation, which this program thinks of as within species error, so we included that. And here are our results. The two best models, so here's the change in AIC corrected for the parameters, differences in parameters. We can see that the two best models fit different parameters of OU and BM in the two different regimes. So remember that sigma squared in these models is stochastic diversification rate, and it increases the among lineage variance and trait means, well, the units are the increase per 200,000 about 2,000 generations. That's the length of the tree that we're fitting. All right. And you can see it's a pretty big difference in diversification rate. Alternatively, we could fit a an OU model with two regimes. And if we do that, we get a slight difference in optimum. The same, well, this one, the best fitting, this is the one that just fits a difference in optimum, right? This is the version of OU that leaves alpha the same in both regimes and sigma squared the same, okay? Those are the two models that do better than all the others. So that suggests that maybe they're really, this is finding support for the idea that, that we have a change in the optimum under OU. All right, here's another thing we can do. Let's look at what these parameter fits are telling us. Now down here at the bottom is what Joseph put on the board yesterday. And this is the little Rosetta Stone that takes you from Bilyeu, O'Meara world to Landyland. Because under, there are two, let's look at this. Under the Brownian motion models, there are two interpretations of sigma squared. Under the genetic drift of the mean interpretation, sigma squared is equal to the number of elapsed generations on the tree times genetic variance in the trait divided by effective population size. So if we've got this, if we know that, if we assume that G, or remember our trait variation is standardized to 1, P, so that this is heritability, right? If we take a modal value from our, remember that survey, which would be 0.4, plug those values in, we can solve for N sub E and see what values we get. The other interpretation of BM is that the optimum moves. Now remember, we're just going to look at the optimum. It doesn't matter because in the data, remember, you get the same results in the radiation for the ornament or the preference. But let's think about the ornament, which is mirroring the preference. Sigma squared under that interpretation is T times the per generation variance in the optimum. When you choose a movement in the optimum, you take a sample from this distribution, and that tells you how much you move. We can solve for the per generation variance in the optimum doing that. And then using the OU models, alpha, as we had on the board yesterday, is T times genetic variance, well that's heritability, divided by the width of the adaptive landscape version of the selection surface, omega plus P. So putting in values for heritability for T, for P is 1, we can solve for the width of the fitness function, analogous to a variance. And that's what we're doing up here. So let's go back and look at our results. From the standpoint of parameter estimation, what we're saying is under a two-regime BM model fit, we get an effective size of 25,000 in regime 1 and 700 in regime 2. Much faster evolution in regime 2, right? And we get a variance under the moving optimum interpretation. We get this value for the per generation variance in optimum, right? Don't really have any empirical, but it turns out that's a very, if you take the square root of that, that's a small fraction of a phenotypic standard deviation per generation, right? Right? Just a small movement. If we go to the OU models, we get, let's see, this is our best fitting one. We get an estimate of sigma of omega of 6,000. So that's pretty large. Under these poor fitting models, it could be quite large. But still, 6,000 is really weak selection. This is confirming what we saw in our simulations, right? But here we're fitting models, getting parameter estimates under maximum likelihood from those fits, and we're saying, oh, we're sort of confirming the general picture we've got, but now we've actually got parameter estimates. And the issue is whether you think that's really a realistic effective population size in over a million-year period. I think it's kind of small, and that's why I'm kind of inclined towards the OU model fit and parameter estimates. Yeah, Roberto. So this is a key part of what we're done, so if you want to talk about this a little bit. So I'm hoping to wrap by 12:30. That'll still give us an hour and a half for lunch. If you're starting to get answers.
So
Roberto.
how did you deal with the standard error of the
Oh,
estimations?
So we also got estimates of standard error, which are reasonably tight, and those are reported in the paper. I just didn't put them here because I didn't want to clutter with. So these are just the point estimates of the parameters. We've got standard errors. We have confidence limits for these estimates. You get that from as part of your output. Maybe you saw it. Did you see that in your output? The exercise you just did. Other questions? Okay. All right, so under 6,000, what does an omega of 6 or 25,000, 6,000 or 25,000 look like? It's a very, here's 9 versus 1, right? So you go out to make this 6,000 or 25,000, it's almost flat. And you could say, well, you're just assuming that it's neutral, but actually that curvature actually exerts and helps bound evolution on really long time scales. But the point is, these values, we have virtually no estimates of the strength of stabilizing selection on preferences. But when any of us think about it, we're thinking about maybe 10 times, as weak as, and here we're saying it's like 3 orders of magnitude weaker than selection on morphology. All right, so okay, so here's our conclusion from this part. Brownian motion of a natural selection optimum is probably the best interpretation. We don't have to make weird assumptions that we've got very small effective population size for millions of years in the one part of the radiation. And that conclusion is consistent with our qualitative results from the phenotypic model exercise, that it would be hard to, you have to have really weak selection on preferences, but we can do it easily with preference movement. And then finally, the Brownian motion of an intermediate optimum may be a reasonable model for adaptive radiations in general. So this result, that just having a stationary optimum and a balance between drift and stabilizing selection with a stationary optimum, in general, you have a very hard time accounting for adaptive radiations, not just in birds of paradise, but wherever it's been fit with morphology. You end up having to assume that you've got only hundreds of individuals in your population, very weak, this is under the genetic drift interpretation, very, very small population or very weak stabilizing selection to account for the model under the Brownian motion of the genetic drift of the mean. So my takeaway from all of this is that in general, pick your process. It could be Brownian motion, it might be BM, but the point is moving the optimum is probably a more reasonable model of adaptive radiations. So for microevolutionists, we don't typically think about a process for moving our optima, right? So this is on a on a macroevolutionary time scale. The data are telling us, you've got to pop out of your microevolutionary world and think about the movement of your optima, and that the movement parameters are going to completely dominate the macro picture. They're going to almost completely suppress the roles of other underlying microevolutionary processes, or at least that's the potential. It's not quite like saying the early part of the course is irrelevant, but you want to temper your posture coming out of the first part of the course when you look at this part and say, okay, well, optimum movement might be the thing. Monique? Oh, you better turn on your mic.
Instead.
Yes, it will.
Instead of optimal movement, could it be interpreted as multiple optimum? Quite
So
maybe
let
close to each other?
So let me say a word about that. In in some depictions of adaptive landscapes for irradiation, there will be multiple peaks. And so you've got a landscape with multiple peaks out there, and the radiation plays out on that stationary multiple peak landscape. That is phenomenally hard to model, and essentially no one ever models that. The closest we ever come is having two adaptive peaks and looking at what are called peak shifts, drifting between the peaks, because you've got to go against the force of selection if you're on a peak. So what we're doing in these in adaptive radiations, generally modeling them with Landy-Land kind of interpretations, is we're having each population responding to its own set of selection. It's got its own private peak. Now there may be a sympatric congener that's evolving, but it's not on the same landscape. It's on its own landscape, and it's following its own peak. And one justification, well, we can model that. That's a huge convenience, whereas the multiple peak thing, basically intractable. But with having private peaks, then you get a radiation by the peaks moving, right? So the movement of peaks and their disposition in trait space starts to look like an ecological problem to me, more than a genetic problem. But it's also consistent with something we found back in the early part of the course, and that is when we looked at beta, which is proportional to your distance from a stabilizing peak, we found that the betas were very close to zero and fell off sharply as you moved away. That's saying that most populations we look at in microevolutionary studies, most trait population distributions are very close to a peak. They're not, it's not like you get cases in which the peak is 10, maybe even 20 standard deviations away, but that's very rare. That's very rare. And that also to me says that the private personal adaptive peak model that we're working with is actually realistic. So.
Could I make a comment about the multiple peak model?
Yeah.
The difficulty is that if the peaks get moderately well separated on a multiple peak model, you end up on one peak, and it basically will take you geological timescales to get to wander enough that you happen to get under the minimum and find the other peak. It's just the same problem you have with Markov chain Monte Carlo methods, where if you have a multi-peak surface and you're trying to sample from it, it gets up on one peak very easily. And as Steve indicated, we don't have good, there are rough mathematical approximations, but just saying that there's multiple peaks may not predict much movement among those peaks unless they're fairly close together.
So in the literature, peak shift means stationary peaks, and you're moving between them. And when I look at those equations from whoever's doing them, basically you need a population size, effective size of 250 or less to get a shift between peaks. So in any kind of even a geological time scale, it takes a long waiting time. So yeah, okay, so we're saying the same thing. I think we've got some general conclusions and then we're done. Okay, so here's my view of the future just from this exercise. And that is, it would be nice to have an analytical solution. Oops, sorry. Oh, you had a question before we go here?
Is there any room for like a character displacement type process at speciation events that would spring things apart?
There is,
yeah.
But within this model, we haven't even got speciation in this model. But we've done other models of this tango with speciation. So I mean, this is a common reaction that there are more elaborate models for speciation, certainly many more elaborate models for sexual selection. But let me just say, we haven't even tested the basic model, right? This is, I think it's almost, I'm not being Donald Trump to say this is the first test of the Fisher-Lande process over the last 30 years on the scale of irradiation. So yeah, so I think what we need, if you can test, sexual conflict and lots of other things, but you need to add them. you need to get this framework up and running, and then you could start adding things on. So that's sort of what you're talking about is a higher order. Yeah, you could. It's of interest, but we're a long way from being able to do that. Okay, so it would be
Steve, one point right there,
scary up close.
is that if you had a sexual preference and sexual trait model, and there were two populations, and they spread out far enough from each other, not quite a Fisher runaway process, but a broad spread, I would think that would predict a certain amount of a certain amount of reproductive isolation.
It does. It does. And we
The speciation process maybe could even involve this.
it could. I call that sexual speciation. And we've written a couple papers about that. We've shown that the same model, then you have multiple preferences. And anyway, so it's in the bibliography. There's a paper by Weda and some of us, 2009? 2009?
Population sees another and says, I wouldn't want to mate with that one.
It's true. But so what happens is you get, under this model, you're drifting along a line of equilibrium with no selection on preference. And so as you drift away, the populations, you really start hating the phenotype of the other population. You get a buildup of sexual isolation. And then you drift back, and so you get the waxing and waning of sexual isolation. So it takes a very long time to drift far enough away that you're not going to come back in contact. Anyway, you can read about that. It's basically the same model, just with multiple populations. Okay, so we need an analytical solution to the phenotypic tango model, I think, but we haven't got it. We need a version of the model that includes peak movement. Actually, I've done simulations with the peak movement, so you'll see that in the paper. We need better data and more of it. It would be nice to have preference data, but maybe we don't need it actually. But I think it would still be nice to have it. And then we need a testing framework that actually matches the biology, and instead of just having to focus on singular pairs of traits on the male side of the equation, and that estimates the parameters. Now, if you go back and think about Joseph's talk about Bayou does a lot of these things. We just need to apply Bayou or expand Bayou so it does coevolution of male and female traits. And then finally, here's an acknowledgement slide. Whoops, what happened here? Here it is. So it's good to interact with your colleagues in general, but here at NIMBioS. And so I got the idea for, using AUI or BM and OU models from attending the course. And as an instructor, I actually learned something sitting out there. So even I can learn here. And so here's an acknowledgement of all the instructors that I've collided with. And Brian helped me with the OU, with AUI, and so did Jeremy. And those were big helps. And I got good feedback from the classes that were preceded this work.